Alrighty. Couple of reminders for you. I am working on grading your intro paragraphs. Thank you to those who have turned it in. There are a couple of you who have not turned those in. I did put zeros in the grade book, so if you have a U right now, that's why. That's the only thing in there. I do take late work, so if you want to still turn that in, I will accept it for a couple points off. Another reminder, I've told you this before, I'll probably tell you again on Wednesday. There's no class on Friday, so please don't show up looking for me. I won't be here. For next class, sorry, this is confusing. For class next Monday, February 10th, please read sections 4.3 to 4.5 in the online textbook. I'm telling you now, you have quite a bit of time to read through that. It's covering organelles. So I want you to read that section in the book prior to coming to class, so that way we can do an online activity. Give me one sec. I just want to make sure that my audio is working for this recording. Okay, cool. Last thing, I am going to post a study guide today for your exam that's coming up in a couple of weeks. It's not fully comprehensive. It is just outlining some of the key pieces that you'd need to know, structure, function, things like that. I did previously mention we'll have a review day, so there's that to look forward to as well. If you have questions as you're working through any sort of like study materials, just let me know. Questions on this? Okay. If you need help finding the textbook, you can also let me know. It is linked on the syllabus. It's free. You don't have to pay for it. You just can follow the link. It's called OpenStax 2E Biology. You don't have to read all of chapter 4, just specifically 4.3 to 4.5. I think it's like, I don't know, 15 pages, maybe 20 pages. For today, we're going to finish up talking about macromolecules. So lipids, proteins, and nucleic acids. Same deal as before with carbohydrates. I want to make sure you're clear on the structure and the function of these different types of molecules. And then particularly with proteins, we're going to be focusing on protein folding and modification. Protein shape, I'm going to talk about a lot today and as we move forward. It's a really foundational biology component. So next class, we'll also have a little activity based on protein structure. So it is something that's not going away. So get comfortable with it, hopefully. So lipids are that category of macromolecules that don't technically fit into our description because they're not technically polymers made up of monomer subunits. Lipids are comprised of other molecules. They're generally too small to be considered macromolecules. So again, some kind of caveats here. This figure is just showing some of the diversity of lipid molecules that exist. You don't have to know all of these, but there are a wide diversity of lipids beyond even those that we're talking about in class today. So lipids, by definition, are hydrophobic molecules. And generally, the reason that they're hydrophobic is because they're nonpolar for the most part. So we know water is a polar molecule, and therefore it tends to dissolve other polar molecules better than nonpolar molecules. So I broke down this word here for you, hydro, water, and phobic meaning fear. So they're water-fearing molecules. As I was just mentioning, that means they do not dissolve in water. They're insoluble in water, which underscores a lot of their biological function. That insolubility in water is key to the way that they function. Their main biological functions are as membrane molecules. So we're going to have a whole lecture where I talk a little bit about biological membranes and how those membranes work. But phospholipids are one of the main, most important molecules in biological membranes. Lipids are also important for regulatory purposes. They're important in the endocrine system specifically. So if you look at the bottom figure here, steroids and waxes, we have like a little ring structure. Those are steroids generally, and those are important for helping different organisms kind of understand what's happening and what needs to go where. They also help in other sorts of insulatory and cushioning properties as well. There's a couple of different categories of lipids we're going to be talking about. Neutral lipids, which are fats and oils. We're also going to talk about waxes. Those are also technically neutral lipids. Phospholipids I was mentioning with membranes, and then steroids as well. So getting into those neutral lipids, those fats and oils, typically they tend to function as storage and insulation molecules. And they're called neutral lipids because they don't have any charged groups at a cellular pH. Generally, these are going to be oil at room temperature. or they will be oils if they're liquid at room temperature, and they will be fats if they're semi-solid or solid at room temperature. So that's how we kind of differentiate groups within this category of neutral lipids or of fats and oils. Now, mostly these molecules are going to function as long-term storage molecules. So a single gram of fat houses a lot more energy than a single gram of polysaccharide, for example. And the structure that I'm highlighting here, the bullet point is talking about the elements that are being highlighted in the figure. So triglycerides are what are being shown here, and they're comprised of a glycerol, which is this kind of orangey square, and then a fatty acid, which is a hydrocarbon chain. Now, the name triglyceride is kind of helpful in understanding the function because tri means three. We have three fatty acid chains attached to that glycerol molecule. So this is the structure of these triglycerides. Sometimes they're called triglycerols. If you see either of those words, it's meaning the same thing and it's referring to this specific molecule. Now, these triglycerides are formed via dehydration synthesis. So we can also see that in the figure here. You see the little water molecule kind of coming out of the glycerol and the fatty acid chain. So that's what we're seeing here. We have a dehydration synthesis reaction linking this fatty acid to the glycerol. This is going to happen 3 times. So in the formation of one of these molecules, we would lose 3 water molecules. Now, one last thing to note here. In this figure, the fatty acids are slightly different, the fatty acids that are being depicted. So we have 2 that are the same and 1 that is different, and that's standard. Within these triglycerol or triglyceride molecules, the fatty acid chains can be the same or they can be different, and that does affect their structure generally and their function, but it's just something to note. Sometimes they're different, sometimes they're the same. These chains tend to be about 16 to 18 hydrocarbons long, so they're not super duper big, but they can be longer or shorter than that as well. The last thing I'll mention here is that generally when chains are longer, they tend to be more oily and generally less soluble. So they're not soluble in the first place, but they continue to be increasingly less soluble the longer the hydrocarbon chain gets. So this image here is showing the formation of a triglyceride molecule. Now this is kind of funky to look at, but the main things to note here, the blue circles are depicting the water molecules and the atoms that are comprised to form those water molecules. This is representing that dehydration synthesis reaction to form this triglyceride. And in this figure, you're noticing maybe the R here. This R is representative of a hydrocarbon chain. So we don't actually see the hydrocarbon chains here, but those R's represent those hydrocarbon chains. I'm going to talk a little bit more about the fatty acids that make up these types of lipids. There's 2 different types. There's saturated fatty acids and there's unsaturated fatty acids. These essentially refer to what types of bonds that they have and what properties they're going to have at different temperature or at room temperature. So saturated fatty acids do not have any double bonds, and we call them saturated fatty acids because they have the maximum number possible of hydrogens bonded to them. They're saturated with hydrogen. So saturated means full to the greatest extent possible. They're full to the greatest extent possible of hydrogen molecules, or hydrogen atoms, I should say. Now, generally, these types of fatty acids, as I was saying, are going to be solid at room temperature, whereas unsaturated fatty acids are going to be oil or liquid at room temperature. And the reason for that is because they contain these double bonds, which cause a kink in the structure. So the chain structure becomes kinked, and therefore the molecules actually can't pack together as closely. So the reason that saturated fatty acids tend to be solid at room temperature is because they're close together, and that's what allows for that property. So they're that single chain and they can get really close together, then they're a solid, but the unsaturated fatty acids have double bonds, which don't allow them to pack quite as closely together. There are monounsaturated fatty acids as well as polyunsaturated fatty acids. That refers to how many double bonds are present. So mono, there's just one, and poly would mean there's multiple double bonds in that molecule. Now, in general, when we think kind of like what organisms have different types of these fatty acids. Generally, animals tend to have more saturated fats and plants tend to have more unsaturated fats. Now, adipocytes are the specific cells where fats tend to be stored in animals. And generally, fats or lipids are stored in the seeds of plants. One thing to note, so the saturated fatty acids tend to be solid and they tend to be a little bit more compact. If we think about why that's advantageous for animals to have rather than plants, animals have to move around. So it's helpful if their energy reserves are more compact and more well stored so that they can move more easily, whereas plants, they don't move, so it's less critical for them to have as compact energy storage. There are nutritional associations as well. You might have heard about saturated versus unsaturated fats on like nutrition labels and things like that. There's some evidence to suggest that diets higher in saturated fat tend to be associated with plaque buildup in arteries. There's some folks who say that's maybe not entirely correlated, but there are some dietary implications here. If you'd like to read more about that or talk more about that, you can let me know. I'm not going to spend a bunch of time on it, but there's a lot of literature on that. So just reach out. This figure, I just wanted to provide you because I think it's pretty helpful in kind of visualizing these differences. I've annotated this with the little arrows. So when I talk about the double bonds, if you're going back and you're looking at these figures and you're like, well, Nora, this is kind of confusing. This saturated fatty acid has a double bond right here. I'm looking right at it. This is a carboxyl group. So I'm actually talking specifically about double bonds within these hydrocarbon chains. So here you can see we have the purple arrow I put on here for you. We have one double bond in this mono unsaturated fatty acid. And then down here, we have an even more kinked chain structure with this polyunsaturated fat. So if you're looking back at this later, maybe this will be a helpful visual for kind of studying these properties. Questions here? So that top one, I'm saying that the double bonds in the carboxyl groups don't relate to double bonds within the chains. Yeah, that's a great question. Yep. So when we talk about double bonds, it's specifically within the hydrocarbon chain, and the hydrocarbon chain are the groups of hydrogens and carbons. Yeah, anything else? Cool. So our next important group of biologically relevant lipids are the phospholipids. These are going to make a constant appearance here. So phospholipids are two fatty acids and then a phosphate group attached to a glycerol backbone. So the structure is honestly pretty similar to a triglyceride. Now, phospholipids, as I said at the beginning, are very, very important for biological membranes. And the reason that they're so critical and the reason that they work the way that they do is because they have this amphiphilic property. They're amphipathic, meaning they have both hydrophilic and hydrophobic components. Now, this figure I've put on here for you because honestly, it's a really helpful figure. It's pretty well annotated. But the hydrophobic part, or the part that doesn't want to interact with water, are the fatty acid tails. And the hydrophilic part, or the part that can interact with water, is going to be the head of the phospholipids. So in general, when you put a bunch of phospholipids in an aqueous or a water-based environment, we tend to see them form these clusters or these lipid bilayers naturally because the fatty acid tails are not interested in interacting with the water. So they're going to face each other, and then the phospholipid part, or the hydrophobic part, is facing each other, and the hydrophilic part faces out. So that's what we're seeing here, which is an image or kind of a piece of an image of a phospholipid bilayer, so a membrane. So when you look at this, again, you might see some structural similarities to the triglyceride. The key difference here, so this is our glycerol, and if we had a triglyceride, we'd have one more fatty acid chain bonded right here, but we don't. Instead, we have this phosphate group. Now, the phosphate group is what can interact with the water. Yep, absolutely. Yep. So the reason that bilayers are kind of funky is because cells have their interior area and then the cells have like the extracellular environment. And both the inside of the cell and the outside of the cell are made up of water. So the bilayer is helpful because the two phospholipid heads can interact with water in both of those environments. And I will talk quite a bit more during our membrane lecture about how that specifically works. But for today, the really key piece is knowing that phospholipids are amphipathic and that they're a really critical class of membranes. I'd also want you to know the basic structure of a single phospholipid. So like that big zoomed in single phospholipid, that would be kind of one of our learning objectives for today is recognizing that structure. It's a good question. Now, because of the structure of these molecules, and this is fairly interesting, I think anyways, if phospholipid bilayers are disrupted, they actually just automatically reform into those bilayers because it's chemically favorable for them to orient themselves in that way. So they're stable when disturbed as a result of their properties. Next up are steroids. So you'll notice that these are structurally very different than phospholipids and fats. They have this fused ring structure. They tend to be 4 fused rings. And this image I have up here for you are common and important steroid molecules. Steroids are almost completely hydrophobic. There is a tiny little part, this hydroxyl group, which is the OH. So this hydroxyl group can actually interact in certain areas with like membranes, for example, but all of this would be hydrophobic. So they're mostly hydrophobic. So that hydroxyl group is the OH group. And these are really important for regulation. Cholesterol sometimes gets a bad reputation, but cholesterol is actually a precursor to other hormones, and hormones, again, help with physiological components. Cholesterol is synthesized in the liver, and it is an important part of membranes. So cholesterol is chilling with the phospholipids, and together they make up biological membranes. Questions here? Last up, we're going to be talking super briefly about waxes. I didn't even put a picture of their structure on here because I'm not going to ask you to identify it. They're long-chain fatty acids with long-chain alcohols. They do play a much smaller biological role. They are relevant, but they're much less important than things like phospholipids in living organisms. Generally, they tend to be used for things like water-resistant coatings. They tend to be solid at room temperature. They can act as a water repellent, or they can act to keep water inside. So a couple of examples I have up here for you of waxes in biological systems. This figure is kind of funky, or the picture of the bee. Bees are able to take sugar and then turn it into wax in special glands, and that's what that is showing. The bee is secreting wax in those glands. I put the beehive or the structure above that they would make with the wax. Then a couple of interesting pictures here. If you own houseplants or you're familiar with houseplants, you might see this. This is a rubber tree. And if you've ever interacted with this type of plant, they have really thick, waxy cuticles. So their leaves are really thick, and that's helpful because they tend to live in hot environments in like their natural systems, which keeps them from losing water as much. There's also an Amazonian giant leaf frog, and they also apply wax around their body. Same deal to keep the water inside, to keep them from desiccating or drying out. Questions here? Mm-hmm.
But white stuff is the actual secretion of the wax.
Mm-hmm. Yep. Kind of weird looking, huh? I also wonder how they took that picture, but anything else? Cool. So I have a question for you. What do these organisms have in common? And I want you to think about this, maybe chat about it with a partner. And then I'm going to have you tell me, what do they have in common? Hey, what do we have up here? What are the creatures? Let's start with that. What's in the top? Duck. Does anyone know what type of duck? It's a widgeon. Um, Below, what is that? There is a beaver and a whale shark. Cool. Yeah, absolutely. So what do these things have in common? And this could be really basic. What do they have in common? They're animals and they live in the water. Yeah, absolutely. Both of those things are true. In terms of like lipids, maybe some thoughts about how these organisms might use lipids. Aquatic organisms very heavily rely on them. Insulation, yep, beavers are totally going to use lipids for insulation. Ducks also do some of that for buoyancy. And if we think about how lipids tend to react with water, this makes sense, right? Because it will keep them warm and dry if they kind of are able to coat themselves in those waxy or fatty coatings. It helps. And then whale sharks tend to have quite a bit of fat on them, and other types of sea animals do as well. That helps with buoyancy because fat tends to float in water. So different properties, but yeah, this is not really a trick question, but it's just something to kind of think about here. Now, one thing that's kind of interesting that a lot of people don't know about, and this is kind of a weird aside, beavers tend to be very oily organisms. If you've ever been in like a museum, or if you've ever seen a beaver pelt, for lack of better words, a beaver specimen, their tails have a lot of fats and oils. They actually have something called castor sacs as well that produce a specific secretion called castoreum. And kind of gross, it was used historically as a food additive. So this picture is showing the castor sacs, and this is not showing anything else. This is showing where you would cut them off. So these kind of gross, these are anal glands, and then the castor sacs above. Now, both female and male beavers have these, and what they do is they're extracted, they're dried, and then they're distilled in alcohol, and it has somewhat of a vanilla flavor. So for a long time, things like Pop-Tarts and ice creams and different sweet treats had castoreum, which is essentially a combination of beaver oil and anal secretions. So it is crazy. Yeah, some perfume companies still use it. Yeah. Uh-huh. How did they find that out? Gosh. Well, I don't know who was like cutting the beavers apart and why they would have eaten it, but I actually don't know the answer to that one.
How did they get the wax all over the body?
Ooh, how do they get the wax all over their body? So they do produce some amount of secretions like from those glands, and then they kind of just use like their feet and their tails to kind of like wipe themselves down a little bit. They also have fur, and fur tends to naturally distribute oils. If you've ever put like flea medicine on a dog, it tends to naturally spread because it's like lipid based. So it goes through the fur a little bit easier that way. Any other questions? Yeah, if you look this up, it's kind of interesting. But yeah, there's lots of foods that used to have this. I should find which specific foods. Okay, here's your break. Using only your memory, I want you to draw a phospholipid and a triglyceride. I don't want you to use your notes. I want you to use your brain. This is just practice. You don't have to turn it in. But after you're done with that, I'm going to have you compare. Don't look at the slides. Just try this. Practice this on your own. Because I do have the answer on here, but try it on your own first. And it can be really simple. You don't have to draw all the carbons and the hydrogens. We can simplify this drawing into like shapes, blocks, squares, lines, circles. If you don't have paper, you can come draw on the board. Who wants to share with the class? I even have a blue marker. Come on, writing on whiteboards is fun. And if you've completed your drawings, check with a partner, see if you got the same thing. Check your answers. Did you get the same thing? If you didn't, maybe discuss who was right, who was wrong. Are you ready for the answer? I guess. Did I hear someone say no? Yes, we're ready. Okay, I should have taken the slide out of here and then made you work for it. Next time I'll remember to remove the answers. So this is a really basic illustration. Here we can see the fatty acid chains are being shown just as like these long rectangles. Obviously, we would have more detail in a figure that I would put on like an exam, but some of the key pieces here, we have a glycerol in both a phospholipid and a triglyceride. The main difference, as I was emphasizing earlier, is the fact that the triglyceride has 3 fatty acid tails, whereas the phospholipid has 2 and then a phosphate. So when we break it down like this, it's hopefully not maybe too bad to kind of think about these two structures. They're pretty similar, but there is that one phosphate group that makes it a big difference.
So on triglyceride, one of those fatty acids, did you say it would be typically would have a double bond?
It's super variable. So these fatty acids could be monounsaturated, polyunsaturated, saturated in any of these different molecules. It varies based on what organism it's in. And this is kind of getting a little bit like lost in the sauce here, but certain organisms have certain like physiological demands. So if you're a fish living in an Arctic Ocean, your cell types are going to be different than if you're like a thermophilic bacteria living in a geyser in Yellowstone. And so the types of fatty acid tails that will be present in different organisms vary. So it kind of just depends. Even within like individual organisms, you could have some fatty or some unsaturated and some saturated. So yes, it varies. And that's a great question. Yeah. Anything else? Cool. So drawing is pretty good for your brain. I know sometimes it can be hard, but there's a lot of evidence to suggest that like practicing drawing really cements concepts in your mind. So that is usually why I ask you to do it. It's not just to like torture you, but something to note. Next up, we're going to talk about proteins. Proteins are crazy. They are very, 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 very important for the functioning of every living organism. They make up more than 50% the dry mass of cells. So if you took the cells and you like dried them out so all the water and everything was gone, more than 50% of that remaining mass would be of proteins. The name protein actually comes from the Greek word proteos, which means first or primary, which kind of underscores their importance. They're just super, super important, very diverse organisms. All proteins are combinations of amino acid polymers in a specific sequence. So amino acids are our monomers and they link up to form polymers. They're very abundant. And as I was just saying, they're extremely diverse in function. On this slide, I've put just a couple of different types of proteins. There are more than this. And the next slide, I also have other examples of protein structure and function. So this list here, I'm showing some of the different things that proteins do. Sometimes they help with structure. They might just provide organization. Sometimes they influence protein synthesis, like making new proteins, carrying substances, storing substances. A lot of your immune system is comprised of really important proteins. Enzymes facilitate biochemical reactions. So more on enzymes later. Now, the casein protein I have over to the right is a good example of a kind of interesting complex protein. We'll talk more about the structure in a couple of slides here, but each of these different colored squiggly lines is representing a different polypeptide chain, so different chain of amino acids. Casein is a storage protein, so that's the protein I put up here for you, and it's found in milk. It's the main source of amino acids for baby mammals. Fun fact for you. This figure, or I should say table, I pulled directly from your textbook. So there is lots of stuff lurking in there if you want to go check it out. You don't need to like memorize this. I'm not going to ask you to recall this information. This is just to kind of show you some of the types of proteins and some of what they would do. So you can check this out on your own time. If you have questions about any of these examples, you could look them up. You could also ask me and I can help you as well. So just an example of some of the diversity of proteins that exist. Getting into the specifics of amino acids, really broadly, they're the monomers that comprise proteins. So remember, monomers link together via dehydration synthesis to form polymers. And that's kind of an understanding for all of our macromolecules, right? Those dehydration synthesis reactions are what form our polymers. Now, all amino acids have an amino group, which is shown here, this nitrogen and double hydrogen combination. And then they have a carboxyl group, which is over here. The key piece to what differentiates different amino acids is what we refer to as this R side chain. The R just stands in for any number of kind of atoms, basically. So there's 20 amino acids that we kind of reference frequently in biology that are the most important and relevant. And each of those amino acids has a different side chain group. So I'll talk more about that here in a sec as well. Now, most proteins are comprised of combinations of about 50 to 1,000 amino acids. However, the biggest known protein is titin, which is related to muscles, and it has like 35,000 amino acids. So that's really, really big protein. That would be huge. A large protein would be like 5,000 amino acids, so 35,000 is crazy. Typically, that little R side chain group influences the activity, chemical activity specifically, that the amino acids and the proteins are going to have. That R group basically determines how the polypeptide is going to function in some ways. Questions here, amino acids. Okay. As I was mentioning, there's about 20 amino acids that are used for all organisms. They differ in various ways, and that's what I've listed up here for you. Largely, we see differences in chemical properties like their charge, their polarity, their pH. Mostly this happens because we see different additions of certain atoms. Nitrogen, oxygen, and sulfur are common. Sometimes they have different numbers of carbon, sometimes their arrangement varies. And then this table I have to the side is showing those 20 important amino acids. And in biology, we tend to represent those by either a 3-letter code or a single letter code. I personally think the 3 letters are a little bit easier to kind of identify, but these are all named. So if you see these 3 letters, those are referring to specific amino acids. Now, essential amino acids are amino acids that humans must have in order to form all the necessary proteins. However, we do not have the capacity to make those proteins. We don't have the capacity to synthesize those amino acids. So 9 amino acids we have to get from our diet, and that's what we call essential amino acids. You have to eat them. If you don't eat them, then that means you might not be able to make all the necessary proteins. If you think of something that is a complete protein, again, kind of going back to this nutrition idea, complete proteins are those that have all the essential amino acids and all the amino acids you would need to form proteins. Classic examples of complete proteins are things like beef or chicken. The combination of rice and beans is also a complete protein. Quinoa, yogurt, those are complete proteins as well. So you can eat those things to get your essential amino acids. This is just, it's kind of chaotic, but you don't need to get too stressed about this. This is just showing the essential amino acids here versus the amino acids that humans have the capacity to synthesize. So the non-essential are the ones that are outlined in a solid line, and then the essential are the ones that have this dotted line. I like this figure because it shows some other kind of differences between these different amino acids. So we can look at the colors. Light blue means they're basic. Yellow is this sulfur containing, etc. So you don't have to memorize this, but it's just a good example of some of the differences in our amino acids. So getting back to proteins, our amino acids are going to be linked together by dehydration synthesis to form something called polypeptide chains. And that's what I'm showing you here, the formation of that peptide bond, which is a covalent bond. So they're pretty strong when they get linked up. And polypeptide chains are polymers of amino acids. So there's a lot of vocabulary terms there. I hope that you can recognize those words. Maybe not today, but certainly as we move forward. So again, polymers are those chains that are hooked together. These chains are made up of amino acids. They're polypeptides. That's their name. Now, the polypeptide chain alone is not a protein. Proteins are combinations of polypeptides that have gone through multiple levels of structural modification to adjust their shape. And this is kind of confusing. So the specific sequence of amino acids is what determines the ultimate shape of a protein. Because the different side chain groups essentially dictate how that polypeptide is going to fold and what its final structure is going to be. So polypeptides fold up to become proteins. Questions on this? And we will review this because it is a challenging idea. There's a lot of like nested hierarchy here. So we have amino acids combining into polypeptides. Polypeptides fold up to form proteins. That's kind of what we have going on here. Now this figure looks scary, but it's really not so bad. I just wanted to show you this because it is looking at a couple of key ideas. So first of all, we have the different amino acids in this 7 amino acid polypeptide color coordinated. So we can see they have their name written out and then their abbreviation. They're also numbered by the specific sequence that they should be for this protein. We can also note here that we have what is referred to as a polypeptide backbone, which is that carbon-nitrogen structure. It's fairly strong. That's the backbone if you hear me talking about that. So nothing too crazy here, but just a good example of kind of what these polypeptides look like. And then now I'm going to talk a little bit about how the proteins get their shape, how they get their structure. So it truly can't be overstated how important the shape of the proteins are to their function. It's so critical. If they're not in the right shape, they won't be able to serve their purpose. And they have to go through different steps of modification before they become proteins. This final bullet I have on here for you, polypeptides do not equal protein. I put that on there to remind you that a polypeptide alone is not a protein. A protein is a protein once it has gone through these 3 or sometimes 4 levels of structural changes. So this sequence is what proteins go through to become a protein. So they start with our polypeptide chain, they get folded up based on their chemical reactivity basically into their 3-dimensional shape. So I'm going to talk about these in a little bit more detail. We can talk about the primary structure of a protein, which is the sequence of amino acids. So again, primary structure is the sequence of amino acids in their specific order. If a single amino acid is changed, we can see changes to the secondary and tertiary structures as well. So our primary structure, and I guarantee I'll ask you about this later, is our chain of amino acids. Okay, our secondary structure is when that polypeptide chain folds up into things we call alpha helices, or an alpha helix as is stated up here, or a beta pleated sheet. So those are just descriptions of what they look like. A helix is like a spiral. So we have like this spirally dude, or we have this foldy wiggly guy. Those are the sheets. Now, typically, what dictates this folding structure is, again, those side chain groups. And these structures tend to be stabilized by hydrogen bonds. Okay, so our tertiary structure is when the secondary structures, those helices and sheets, are going to combine together, and parts of them are going to be facing outside and parts are going to be kind of hidden. Now, when we talk about tertiary structure, This is essentially the three-dimensional shape of a protein. This is where it really becomes a protein. Charge side chains, van der Waals interactions, ionic bonds are all important here. And some bits of those side chains, again, are concealed and some are revealed intentionally. The tertiary structure also influences the solubility of a protein. So all proteins are going to have primary, secondary, and tertiary structure, and some will have this quaternary structure. There's lots of different proteins that are combinations of polypeptide chains, and if they have to combine, that's what quaternary structure and modification is going to be. Those different polypeptides are going to be bound together, and generally we think of these as like larger, more complex proteins if they have this quaternary folding or this quaternary structure. So they don't all have that. Questions here? And next class I have paper models, which are kind of fun. We're going to practice this idea. So I'm going to ask you to identify primary, secondary, tertiary, quaternary structure of the proteins next class. We're just going to be working with 2 polypeptide chains, but this could occur with lots and lots. So this is a good example of how important the primary structure is to the ultimate shape of the protein. Now this figure is showing what normal hemoglobin proteins look like compared to sickle cell hemoglobin. Now, sickle cell disease you might have heard of, which is essentially a disorder that can cause issues with oxygenation. We also see issues with blood clotting, and there tends to be fatigue, pain, or stroke, also vision complications as a result of the malformed hemoglobin proteins. So normal hemoglobin is going to have glutamine in this 6th spot. And as a result, we get hemoglobin that does not associate with other hemoglobin. As a result, it's able to carry oxygen really well, and the body remains oxygenated effectively. However, sometimes, and usually this is genetically influenced, we have a substitution here at this 6th spot, and instead of glutamine, we get valine. When that happens, the entire structure of the protein is changed, and as a result, we get these sickle subunits. So basically, that means the protein is shaped incorrectly, which is why we call it a sickle shape, because it is kind of bent like a half moon. And those sickle proteins tend to aggregate, and then the body doesn't function as well because the oxygen capacity is greatly diminished. It's reduced a lot. One other thing to note about this figure, if you're looking, hemoglobin does have quaternary structure, so it's a relatively large, complex protein. So that's what's showing here. But going back to the large idea, a single change in the amino acid sequence results in really large consequences that are lifelong for some circumstances. So the primary structure is really important. Questions here? Now, one thing I don't think I explained super well, I mentioned that normal hemoglobin is not going to aggregate, which is why it flows normally through things like veins and blood vessels and things. The sickle cell hemoglobin aggregates, and what they kind of call it is it gets into these sorts of fibers, which is what causes some of those issues with like vision. Sometimes it can block the veins to the retina, which is why you have vision problems. And then similar blockages is what would result in like strokes. So it physically is like bigger, which is what causes problems. Okay, I have a break for you. This one's silly. You don't have to think that much. Is cereal soup? Debate with a friend. Again, kind of silly. Kind of silly. What do you think? I think I heard a lot of the same answer, but what do you think? Is it a soup? Yes. Wow, lots of yeses. No, lots of no's. No. Does anyone want to justify their answer? You don't have to. Liquid plus solid eaten out of a bowl with a spoon equals soup. What about oatmeal? Oatmeal is like hot cereal.
Yeah, oatmeal's hot.
Oatmeal's hot. I don't really think oatmeal's cereal personally.
It's a cereal grain.
It is a cereal grain. Or like granola. Challenging debate here, people. We're gonna keep going, but if you want to challenge your side, I'm all ears. Come to my office hours. I have them today after class, 11:30. All right, last up, we're going to talk about nucleic acids. Nucleic acids, we will spend a lot of time talking about in great detail. So a pre-warning for this, I know we only have like 3 more minutes left in class. This is very basic overview. I have an entire lecture about DNA structure and function, so this is really brief. Nucleic acids, both DNA and RNA, you might have heard of. They're very important to storing, transmitting, and expressing hereditary information. That's all that they do in a lot of senses. RNA does lots of other things, but DNA specifically is for storing and expressing this hereditary information. Nucleic acids are macromolecules, so they do fit our definition according to that monomer-polymer situation. And we have 2 types, DNA and RNA. Now, nucleic acids are long polymer chains of nucleotide monomers. Now, this whole figure right here is a nucleotide. So this would be one nucleotide, and we would have lots of nucleotides linked together, again, via dehydration synthesis to form our nucleic acids. Now, one of the struggles with learning about nucleic acids is kind of the fact that we have lots of words that sound similar. So you'll notice nucleic acids are comprised of nucleotide monomers, and nucleotides are made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base. The nitrogenous base varies, and the sugar varies based on what type of nucleic acid we're looking at. So if we're looking at deoxyribonucleic acid, then we're going to be looking at a deoxyribo sugar. And if we're looking at ribonucleic acid, which would be RNA, we're going to have a ribose sugar. So on the next slide, I do have a picture showing the differences. It's very minimal. It's pretty much just, does it have one more oxygen or not? So these are very similar sugars, but I do want you to start getting these different words clear in your head. I promise, I've already written the question. On your exam, there's a question about differentiating nucleic acids from nucleotides from nitrogenous bases. So I want you to be clear on like what those different things are and how they relate to each other. So nucleic acids is like our big category, right? And then they're comprised of nucleotides, and nucleotides are comprised of nitrogenous bases. So if we need to review that more, we can. As promised, here's a figure showing the differences between our 5-carbon sugar. We have our ribose sugar and our deoxyribose sugar. The little red box on here is showing the difference between these types of sugars. It's just one oxygen. I'm not going to spend very much time on this, but there are different nitrogenous bases. Basically, there's restrictive pairings because there's limited space within these types of molecules. So thymine, cytosine, and uracil are slightly smaller molecules, and then adenine and guanine are slightly larger. So we tend to see pairings of like one large, one small, which would be purine and pyrimidine pairings. So this is my last slide for today. Well, I do have this question slide, and we can come back to this for next class. I already mentioned this. Nucleic acids store heritable information, and DNA and RNA together confer gene expression through protein synthesis. So if you have some time before next class, fill this out. I know I already asked you about questions, but I want you to fill this out. There's a link on Blackboard. Do this before next class, please. Thank you. Good day.